Welcome everyone to our webinar in the series From Lab to Life, Translating Science into the Real World Impact. This is the first webinar in the series on biobanking. Now I would request Dr. Elango to speak few words about the vision of our organization, and then we will play a short video on our innovative product of biobanking, and then give an introduction about the speakers of the day. Over to Dr. Elango.
Thank you, Dr. Jiny. Migene is a spin-off company from Neom Technologies Private Limited. And here we present InstaPreserve, which is a recent and innovative model, a substance for storing the specimens at room temperature. In that process, we have started a series of webinars to promote biobanking. The reason behind is in India, the biobanking is in its primitive stage. Only very few centers have come up with the biobanking, understanding the need and the necessity of having a biobanking for any field of science. It could be a veterinary or agriculture or human. So if we have a series of lectures in biobanking, it will create an awareness and increased knowledge in biobanking, why any clinical or any research center should have a biobanking in their premise. It's very, very important to have the biobanking. Any researcher, if you ask, for their study, they collect the specimens. The biospecimens could be leaf, stem, root, soil, or milk, or veterinary samples. In human, we collect blood, saliva, urine, body tissues, and body fluids. But we collect the samples and store, do the research, then we stop it. For any time, every time for a clinical study, we keep on collecting, we spend considerable amount of time in collecting the samples. So more than 30% of the time it's spent on sample collection. If you have a biospecimen, a biobank in the premises, that 30% time can be saved. I'm sure that the webinar series which we have initiated now, this is the first seminar, and we have a right person, Dr. Amritha Suresh, with us. She has established 2 biobanks, one already in Amritha Hospital in Cochin, and now the biobank at Manzumdar Shaw Translational Research Center in the premises of Narayana Health City in Bangalore. We have video. So before we get into the web series, our webinars series, we would like to have a small introduction of our product and the company. This product has come out of the pain as a researcher which we went through earlier. And the innovator is Dr. Rajinikanth Vangala. For want of money to store the samples in the freezers and liquid nitrogen, when the fund is depleted and there is no fund further to keep the samples, with no other option when we have to throw the samples in the drain, that's the real painful for any researcher who cannot afford it. So out of the pain, when alternate solution is required, and Dr. Rajinikanth Vangala has come out with this model of an InstaPreserve where he can collect, transport, and store the biospecimens in InstaPreserve. We'll have a video on Migene with the product description.
Recording in progress.
Now we have Dr. Amritha Suresh with us. She is a well-known leading research person in the clinical settings, and she is the principal scientist of the Integrated Head and Neck Oncology Program at Manzumdar Shaw Translational Research Center, which is situated within Narayana Health City, Bangalore. She is also a faculty of Roswell Park and Manzumdar Shaw Collaborative Research Program. She did her doctoral thesis from a premier institute in India, the Center for Cellular and Molecular Biology, and she joined Amritha Medical Sciences in Head and Neck Research Institute as a research associate. Then that is the time she was setting up a research facility and head and neck tissue repository. And I was so happy to inform that even that period, we were associated in setting up the tissue repository. So I know personally how much effort she has taken to establish the biobank from the scratch and also have its own software. She very well managed, and after joining here in Narayana Health City, the Manzumdar Shaw Center for Translational Research, now she has established a full-fledged biorepository for head and neck cancer research, wherein the research activities are starting from genomics to proteomics, from salivary samples to blood samples to body tissues, up to the clinical research. So I'm very happy to share that Dr. Amritha Suresh is the right person to begin with in the series of webinars on Biobank. And I am sure for everyone, every researcher, this will open a new window in their thinking process of collecting and storing the biospecimens. So I welcome Dr. Amritha Suresh. I'm sure this will be a successful beginning for creating an awareness and the collaboration between the multiple centers in their respective area of research interest. I'm sure this will contribute to a large extent to the individuals, to the institutions, and on the whole, to our country. And over to Dr. Amritha Suresh.
Thank you, Dr. Elango. I hope I'm audible. Hello? Hello?
You have to share.
Hello, am I audible?
Dr. Amritha? You have to unmute. Yes.
Am I audible now? Yes, okay, sure. Unfortunately, my video is a bit of a problem, so I guess we can continue with the audio setup itself, right?
Yeah, that is okay. That is okay.
Yeah, that's okay. Just a second. I'm just sharing my screen.
It doesn't get
Is my screen visible? Hello?
Okay, yeah. Your screen is visible.
All right, okay. Okay. Thank you very much, Dr. Elango and Dr. Rajnikant and the NEOM team for the invitation. So what I've been requested is to speak on biobanking and being the hidden treasure, unveiling the untapped potential, and how it is so relevant for research in clinical as well as basic science research. Thank you once again for the opportunity. So biobanking is currently a concept that is very much increasingly in focus. If you take the current literature across the years from 1996 to 2019, there are n number of papers that have come out in the field of biobanking, and there's a gradual or an exponential increase in terms of the work that is being done in terms of biobanking. And the publications are mostly in terms of various aspects of biobanking, ranging from the disease conditions to ethical considerations to public health, personalized medicine. So there's no doubt that biobanking is one of the milestones that are required if anybody who's in vision, who has a vision of doing any kind of research in terms of clinical or translational. Now, what we would do in the next, say, 20 to 25 minutes is to look at biobanking, the basics of biobanking, the components that are involved and the guidelines that govern it, and the translational implications. So when you talk about biobanking, there are a lot of topics that come out in connection to it in terms of specimen storage, the kind of applications that you can do, the QA and QC that is required, the ethical considerations, the logistics. So there are a lot of topics that come into everyone's mind when we talk about biobanking. We would try to delineate a little bit of them during the next few minutes, and then see whether we can have a more simpler understanding of what the biobanking requires. So the need is very evident. The researchers and clinicians have been collecting and studying tissue and information about health and lifestyle for many years. But when you make that a very well annotated, archived, and a maintained biobank, that is what is almost an invaluable resource for advancing understanding of health and disease, to advance development of healthcare, for precision medicine, basic and discovery research, as well as for any kind of innovations. So there is no doubt about the need of biobanking. And there are many names that have been called off, but ultimately what is required is a very well annotated system which can help take research forward. So if you look at a definition-based understanding, it is an organized collection of resources that can be accessed to retrieve human biological material and data for research purposes. Now, they can be bioresource based. So that means it can be based on the type of biospecimen that is connected. It can be a prospective collection. It can be retrospectively collected. It can be surgical specimens. Stem cell banks are there. And although we do have biobanks of non-human materials, I think in this talk, we would basically focus on the human-based samples. Now, we also have dataset-based repositories where you have an organized collection of data and information, whether it can be in the physical or in the electronic or the digital form, that can be used for biomedical and health research. Disease registries, health surveys, disease surveillance data, census data, all of these form a basic biorepository that can be used for data management and further observational research. Even personal health records in healthcare institutions have a very huge potential for subsequent research, which is not very tapped area. Now, when you're doing biobanks, the collection is usually either population-based, which are very, very large-scale prospective sample collections, or it can be very patient or clinical-based, focused on very disease-specific categories such as cancer. Now, what are the key concepts of biorepository maintenance? Now, the important concepts are, say, how do you govern it? What are the ethical and legal issues? How do you manage the data? And actually, most importantly, what are the quality assurance criteria that you would take in consideration? So if you take ethical considerations, you need to have IEC approvals, consenting and confidentiality being maintained. Very electronic data management systems are required. Quality assurance needs to look into processing, storage, annotation, and specimen quality. And governance is basically the whole delegation, specimen maintenance, and distribution, and who takes care of regulating the entire biorepository. We will look into a bird's eye view of all of these concepts today. So systematic collection and storage of high quality samples with the associated clinical data from patients after regulatory approvals and accurate consenting. This is what is the baseline of any kind of biorepository that has to be maintained. So what is a workflow of a general biorepository sample collection process? So if you might have a patient or a volunteer or a subject from whom you need to collect the samples, what we do first is the informed consent, and then the data collection in terms of the demographics, the clinical, the pathological, the risk habits. Then we look at enrollment in terms of the specific, either in terms of a blanket repository or in terms of a study-specific requirement or a protocol-specific requirement with regard to the inclusion and exclusion criteria of the patient, of the subject. And then procure specimens based on specific standard operating protocols. And the specimens can either be tissue, body fluids, saliva, body fluids including saliva, blood, urine, bone marrow, CSF, and also look at their specific storage according to the standard operating protocols. And currently, we also have the addition of development of patient-derived cell lines from the tissues being included in the repository process. All of them being barcoded and cataloged. And then look at Open Specimen as one of the softwares which are used for data storage, tracking, retrieval, and distribution. There are many other, but basically an electronic record for all of these is required so that you have a systematic maintenance and distribution of the samples. Now, in all of this entire workflow, you have specific considerations that have to be taken into account. That is ethics while the collection sample process, enrollment of the patient in the study, it's all the ethics and the regulatory concerns. When you're talking about procuring the specimen and the body fluids and the storage and the barcoding, that is basically quality assurance that you need to take care of. And finally, when you have looking at an electronic data management system, that is essential for the digital management of the data storage, tracking, retrieval, and distribution. So when you look at the first set of guidelines, that is the ethical and regulatory guidelines, in India, the ICMR guidelines released in terms of the national ethical guideline, they are the most followed guidelines, so they are the ones that are required to be followed for people managing the biobanks, which indicates that there is a requirement for ethical committee approvals. Informed consent is required. Now, the challenges with informed consent include that when you're collecting samples for a repository, the study that is required or the aims of a scientific study is not usually decided. You are usually collecting the biorepository as a blanket study. And therefore, there is always a time lag between the collection of the sample and its use in research. So usually the challenges that are brought to because of these issues are kind of addressed by using a multi-layered consent. That means you're basically informing the patient that this might be involved for a certain amount of research. You're taking a broad-based consent from them. You might have a secondary consent when the patient comes in for follow-up to be taken if the the sample is being used for any other purpose than it was initially consented for. So there are ways of going around the process, but basically an ethical committee approval and a multi-layered consent is probably necessary. Now, there are other issues that are addressed in the ICMR guidelines, that is in terms of the ownership of the biological samples and the data, which is basically in terms of who's managing the data. The ownership can range from the patient to the biorepository management to the the sponsor for the repository. The material transfer agreement in case the samples are going out of the institution within the country or outside the country. Secondary or extended uses, as I mentioned, in terms of taking a re-consent from the patients, and the dissemination of the results. I mean, basically to the contributors who are working in the study, to the donors, and to community. In case of the donors, one of the major ethical concerns is to take consent from the patients that when the sample is being collected itself, that whether they want to be informed about the result of the studies that is going on with their samples. And of course, to the community in terms of sharing the results so as to disseminate the scientific knowledge that is gained from the repository. And benefit sharing is mostly in terms of the point in ICMR is mostly in terms of commercial benefit, that suppose you are developing a cell line, suppose you're developing an assay which turns out to commercial benefit, who are the people who would be sharing the benefit? That is something that has to be decided prior before you embark upon the process. So the premier committee that governs all of this is basically the Institutional Ethics Committee, which plays a key role in the oversight and the use of the bio and data repositories for research and in terms of distribution of the data. So they should all be reviewed and approved by the EC. Now, what are the biobank components that are involved? So obviously you have sample collection, you have an analysis for the QC of the samples, you transport it if it's required across to, if it's a centralized repository, then you transport it across to the repository, the preparation and the processing, the storage, and the data management. Now, there are multiple types of biorepository samples that can be collected. The most common of them are the pathology archive in terms of the FFP blocks or the pathology blocks. You do have pre-existing and prospective collections, as we mentioned before. The other most common things that we look at is the surplus diagnostic samples, the surgical samples, surplus diagnostic samples, as well as the surgery samples in excess which come out of the operating theaters. Those are the ones that are stored in kinds of a repository. And of course, if you have a tissue microarray. Now, these samples are then used for multiple services in research like sectioning and staining right up to TMA construction. Now, the fresh surgical sample, now usually if you already have a standard protocol that has been approved by the AEC, then you have prospective collections where the surgical samples are collected, where the collection protocols are standardized. And if it's a multi-centric sample collection, then all these protocols have to be standardized across all the sites. Now, the workflow remains similar, where you take consent, you conduct the surgery, take out the tissue, and then the specific methods of storage. And if it's either whether it can be frozen or whether it can be fixed. And this is a similar protocol. The problem that, or the aspect that we want to emphasize over here is that at every step, now you're looking at informed consent, you're looking at sample collection, you're looking at processing and storage and banking. At every step, you do need standard operating protocols that would decide that the quality assurance is maintained at every step in the process or in the workflow when a biospecimen moves from the patient to the storage system or when it's being cataloged into the software. Now, where is the governance and the management? So what is suggested by the ICMR is a technical committee for a biorepository along with the EC, and who should consist of people across the forum of the users as well as the people who govern it, which should be part of the committee, and they should govern the collection of the specimens, disbursement, as well as the data to the researchers, and oversee the execution of the MTA or the data transfer agreement for transfer of biospecimen or data to other institutions specifically. Now, if it's usually the committees are required for huge repositories wherein there have to be independent technical authorization committees as well as ECs to undertake the above mentioned tasks. The biobanks should have very well-structured SOPs and clear guidelines for collection, coding, anonymization, storage, access, and a contingency plan in case of any troubleshooting that needs to be taken care of. So when you're collecting the data, so at the initial stages, when your sample collection is happening, you collect the data, and there the assurance or the quality assurance that needs to happen is in terms of collecting accurate data, providing preparation of the data forms which are accurate in terms of the specific project that you want to do, or in terms of the biorepository, and in terms of collecting data right from the patient history to the treatment to the outcome, and also in terms of their follow-up data that needs to be included. Now, if you are transporting, now you have, as we mentioned, the sample scan specimen can be of multiple types. It can be right from the tissue to the biological fluids and to cells. Now, each of them have very different transportation and optimal and storage conditions. I'm not going into details of each of these storage conditions, but ranging from the system that was just now presented in terms of the preservation of the samples, you need those kind of systems for tissues. You need a live kind of medium for cells, which need to be viable at a later stage. You need to have systems where preservation of proteins and RNA is required. So based on a specific standard operating protocols, if the transportation needs to be maintained. And once this is done, then there is a systematic archival and documentation of the process by barcoding of the samples and entering into the system, wherein all these informations that are collected are entered into the software, and which is a queryable software wherein data can be retrieved at a later stage. And finally, all the processing of the sample has to be done as per the standard operating protocols and entering into the data management system. Now, if you ensure this entire process of maintaining QC across all these different stages of the journey of a biospecimen from the patient to the storage repository, then we can ensure that there is an accurate and data output that's coming out of it, data that can be used and that can be relevant clinically as well as translationally. So a regulated stepwise process across all these steps would ensure ensure quality control. Now, the important factor is how do you use this data for a basis of translational research? And how is it, the accuracy of the data is something that is the basis of translation. Now, we use it for characterization of patients and samples. We use it for identifying suitable patients and in terms of inclusion and exclusion criteria for your different studies. They facilitate clinical trials. So it is very obvious on the basis of the application of the biobank that there is a essential need for keeping it accurate. But at the same time, this is the major challenge that we find in terms of quality assurance is very essential, but it is a major challenge. Now, everything that you do in terms of translational research, it depends upon a high quality specimen that you get. And considering all the outputs that you actually want the high quality specimen. But our challenge is in terms of, there are multiple variables that happen when you are pre-acquisition of the sample and when there is a post-acquisition of sample. Now, when pre-acquisition is mostly patient-related or subject-related in terms of the subject habits, in terms of the drugs that is being given to a patient, in terms of the anesthesia protocol, the pressure variations, any fluids that have been administered to the patient, pre-existing medical conditions, the patient gender, ethnicity, all these things are the variables that are pre-acquisition variables that kind of determine a certain amount of quality of your specimen. And in terms of post-acquisition, in terms of your temperature, whether it can be in terms of the storage temperature or your room temperature, the kind of fixatives that you use, the rate of freezing that you're kind of doing when you're doing a fresh frozen sample, the kind of processing. Now you have multiple hands handling the sample, the processing, and if the SOP is not followed, then you have problems in the output analyte that you get. The biomolecule extraction methods for whether it is DNA, RNA, protein, you have multiple extraction methods which are available. And if there is no standardization of the process that is being carried out, then you do have, again, different variations in the kind of biomolecules that is extracted. And the duration. Now, storage duration is one of the most challenging factors of the repository, wherein, we do have samples collected from years before, and trying to maintain their quality and control and the type of bioanalyte that is recovered from the sample is a big challenge. Now, the other challenge in the biorepository, or something that is recently come into focus, is the tumor heterogeneity. Now, heterogeneity, this is specifically in terms of cancer-based repositories, where we find that right now the basics research or the translation research does not just focus on the epithelial tumor cells or only the tumor cells independently. They focus on the entire tumor as a whole. But at the same time, when you take a sample for the repository, it is almost never a representative of the entire tumor. So there's always tumor heterogeneity in terms of the percentage of tumor, in terms of the contribution of the stromal cells. And so that is a challenge that has not been that is currently being taken care of. Now, recently study which was published in 2019, they have tried to implement or try to address this challenge by including cell culture biobanks, which are patient derived, use PDX models, that is patient derived xenograft models, and look at all of these together as a combined and comprehensive repository for the patient sample, rather than just look at biospecimen or the tissue or the the body fluids that are collected at a single point of time. So what is being suggested by this paper that was published in multiple cancer types was that all the other factors are similar, like what we discussed. They collect all the patient details, they do the pathology reporting, they collect the fresh frozen tissue, but the additional, if you can see, is the addition of the PDC models, that is the patient-derived cell models, and viable frozen cells and tissue. So they look at the different types of, they collect cell-side cells from the patients at different time points, do the patient-derived cell derivations, and then look at their genomes and RNAs and the proteomes, and then see whether they can kind of contribute towards the cellular heterogeneity. A similar study that was done in breast cancer heterogeneity, in order to address a similar tissue of tumor heterogeneity in breast cancer, they have looked at population-based studies wherein they've collected the patient samples. Then they've looked at patient-derived. The same sample has been put into the mice to develop the patient-derived xenografts, and then the same sample, both from the patients as well as from the xenografts, have been used to develop patient-derived cells. And these have all been profiled, all of them are being profiled, and they all kind of say that both the breast tumor, the PDTX, and the patient-derived cells, they all share a certain amount of intratumoral heterogeneity. So these are kind of the methodologies that have been adopted to address the process of intratumor heterogeneity in breast cancer. So this particular paper has suggested an integrated platform combining the xenografts and short-term cultures as a better way of addressing the problem of intratumoral heterogeneity and the samples in the repository not being representative of the total tumor. So this, in combination, if you see, it can be here. This is something that is for the future, where you can actually probably see that this can improve the repository, the representation of the repository of the particular disease type that is under investigation. Now, just to give, since the whole idea was to understand what is the potential of the biorepository and how it can be applied translationally. In the next few slides, I would just give you an idea about the journey that we have had here in establishing the repository that is currently a combined repository between the Manzumdar Shaw Medical Foundation and Narayana Health, and what are the few studies that have come out based on the cells or the samples or the tissues or the images that have been stored in our repository. So we started a repository around, say, 2009 onwards, and we have more than 4,000 patients and more than 25,000 samples in our repository. And we collect both, we do a multidimensional collection in terms of biospecimen, which includes body fluids, tissue, and cells. And in terms of radiology images, we look at CT, MRI, and USG images for varied disease conditions and for varied contexts in terms of the clinical questions that are being addressed. And histopathology, both FFP blocks as well as cytology samples. Now, since we are working in head and neck, cytology is of a big deal because we do brush biopsies as well as FNACs for thyroid samples. So all these samples together, all these specimens together, and this is notwithstanding the clinical information that is collected while the patient samples are being collected. So using all of these, the biobank-led research program, say, specifically focusing on the head and neck cancer department, because as that is my area of work. Now here, we look at discovery and validation using these biosamples. We do targeted gene validation as well as multi-omics analysis. And we look at susceptibility screening, early detection, prevention, treatment management, prognosis, as well as metastatic progression. So we look at all of them, and we look at developing biomarker panels, point of care assays, and novel targets. Now, if you look at this entire area or the field of head and neck cancer that is being addressed, it's pretty obvious that the clinical data collection at every stage in terms of what is their baseline data collection in terms of the follow-up, and what is their treatment outcome. Everything needs to be recorded extremely, diligently in order to get properly and clinically relevant data. So to just give you an understanding, what we do use is a multi-pronged approach using biospecimen, body fluids, images, patient-derived xenografts, patient-derived cells for doing the research in head and neck cancer. So we have imaging in terms of OCT, molecular imaging, autofluorescence, and digital pathology. We use preclinical models, both in vitro animal models and 3D germa models, and and considering that the number of data that is so extensive, use of AI becomes almost a mandatory. So AI-based automation is another factor. And for all of these things, we use the development of point of care assays to identify patients with susceptible lesions, to non-responders to therapy, and for patients with poor prog. So basically triage the high-risk patients appropriately so that we can provide them timely treatment management. Now, one of the first examples that I would give is about our early detection program. So we have screened a lot of populations which are at risk, and with the screening was done with oral probe, which is being attached to the mobile phone. So we have captured about 30,000 images. And with this is autofluorescence-based imaging. I'm not going into the results of the studies. I'm basically trying to emphasize here the importance of the repository, whatever it may be in terms of the data as well as the specimen, and how the results have been derived from the biorepository. So with these 30,000 images, we had developed an AI-based algorithm which could identify the high-risk lesions with high-risk patients with susceptible lesions with more than 90% accuracy. Then we looked at oral cytology as a method, wherein we used FFP sections of more than 300 and cytology brush biopsies of more than 300 for developing a telecytology-based system, and then using AI to basically classify the patients into high-grade and low-grade lesions. We added molecular markers to this, which was done in another 250 patients, and so that we could classify the patients between high-grade and low-grade with even higher specificity and sensitivity of again around 90%. So these are all technologies which can be applied at different stages. As I said, for the screening, we use the imaging, which does not need any kind of laboratory input, and the output can be given only by AI, and it can be given remotely. The telecytology can be used in primary health centers. It is a small device that can be used. And the molecular cytology needs a little bit more, it's more laboratory intensive, so it can be done in tertiary healthcare centers, but it gives you a more accurate classification of the high-grade lesions. Now, in all of this, the significant QC measures that were taken for using the data that was stored in the repository was accuracy in clinical data. Now, in most of the cases, in multiple cases, the data was collected by using an app, and the data was collected by frontline health workers. So we had to make sure that the frontline health workers were trained in terms of collecting the data and in terms of the image acquisition, that they are actually taking the right image of the lesions. And the training program was carried out every 3 months so that the frontline health workers, their knowledge is refurbished, and there is no, big gap in the quality of the data that is obtained. And in terms of the image quality, so there are softwares that can give you as to what is the image quality what is the quality of the images that have been captured. They were used to make sure that, the data that is going into the AI algorithm are all of a similar QC. And when you're talking about cell numbers, right from the beginning itself, you have to make sure that the cells that are collected are of enough number. You are doing the brush biopsy sampling in the right way, because otherwise what you're getting is just exfoliated cells, which is only right on the topical surface, and you're not going deep inside the lesion. So in the case of FFP sections, FFP sections are being taken from our pathology, so they follow a standard protocol, but the storage in formalin, the time period of storage in formalin, the sectioning that is taken, the thickness of the section, all of that kind of determines your endpoint result. This is in terms of the early detection program. Now, we have carried out molecular profiling studies where we have developed biomarker databases for drug resistance. Now, this was carried out most in laryngopharyngeal cancers. It was a project from DBT. Now, here the problem in this case was we are doing it in a biopsy of the laryngopharyngeal cancer. So the sample is very less. So you get just a 5mm biopsy sample from here, and we had to do the entire multi-analyte processing. So here we standardized the process of the analysis of multi-omic profiling, both transcriptomics as well as proteomics from the same sample. So because of the less sample that was obtained. And here the correlation with molecular data in terms of treatment outcome and response assessment was very important. And in terms of collecting the data at 3 months response as to after the imaging or after the chemotherapy is given, you take the RECIST criteria and kind of understand whether they are responders and non-responders. And here the storage of the sample also created a lot of provided if it's stored accurately, provided the necessary input in terms of the RNA, the DNA, and the protein that was actually isolated. So this was one of the multi-omic studies that was carried out, which gave us a lot of insights upon what is the requirement for the storage of the sample that can be used across different studies. Now, another point here that another project that I would like to mention is in terms of nodal metastasis. In terms of sample collection, this was another challenge, because here we are talking about, so the study was to understand the different mechanisms of nodal metastasis. And here the idea was to collect samples from the node. Now, samples from the node cannot be just directly collected from the theater, because it needs to have a pathological input. And it's only the pathologist who can tell us the sample that can be given to us without compromising the patient diagnostic protocol. So there had to be a standard operating protocol established for collaboration between the clinician, the surgical procedure, the surgeons in the theater, then the examination by the pathologist before the sample is given to the research team. So this entire thing had to be established so that ensure that the tissue is collected optimally, that there is no compromise to the patient diagnostic process, and that the sample that we get is the right sample in terms of the metastatic deposit in the lymph node or in terms of the lymph node stroma. So this was one of the challenges that we had, and that is relevant, that kind of emphasizes the need for optimal sample collection. The other thing, as we said, was developing in vitro cell line models. So we have taken lymph node samples, and with the same protocol that is just now mentioned, we have taken the samples and developed in vitro cell line models. And in this in vitro cell line models, now here the issue is obviously from the tissue. When you take the samples, when you take the cells, it is a heterogeneous sample. You get multiple different types of cell types. So when you develop a cell line, it's essential to establish the cell type specificity. What is the kind of cells that you have got? Make sure that the STR profiling of the cells matches to the patient so that you confirm that it is of the same patient origin, and the purity and the heterogeneity, so that all this is essential if the cells are to be viable enough, or there have to be some relevant data that you get out when you do a proper research out of this. So this is another example of wherein tissue and cells were collected from the same patient, and currently studies are ongoing in this area. Now, another study that happened was which was a challenge in terms of sample collection, was to establish biomarkers and surgical margins. Now, in this case, we had collected samples from different sites of the tumor at different distances of the tumor, and obviously with complete ethical committee approval. And we had looked at different cell markers by FFP at different stages. Now, here the most important factor was to correlate with the clinical data, with the histological data, the annotation in terms of the patient's clinical stage, the pathological stage, the follow-up, and the treatment outcome. So at the end of it, once we collected about 234 samples from about 40 patients, then we were able to understand that the markers could clearly delineate the surgical margins between N0 and N plus patients, and not really only in the N0 patients and not in the N plus patients. So that kind of a result which we get was not possible unless and until we did a proper collation of data and a proper correlation. Now, a last example that I would like to give is a proper bench-to-bed translation, which whatever work that was done in the bench finally led to a clinical trial. Now, we had carried out an animal study where we had taken, this is an animal study wherein we had taken the foreign cure model And the foreign cure model is wherein you give drugs to the animal and it develops oral cancer lesions. And these samples, this is an example of samples being stored from animal tissues, animal models, and these tissues were stored. And then we did the necessary analysis in terms of the stem cell development and in terms of the stem cell markers. We, from the same patient, from the same animal tissue, we developed the cell lines, and those cell lines were then used for looking at what is the role of curcumin and metformin in the chemoprevention treatment. Now, this combined result from the animal model as well as from the cell line derived from the animal model has led to the SPT trial, the second primary prevention trial that's currently ongoing. And this has been coordinated by HNCOG and NHF. Currently, it is recruiting in almost 18 centers, and it has a patient number of 1,500. So this is an example of how, work which is carried out in the bench can take place to the bed. And herein we had the this thing of storing samples from the animals as well as developing cell lines. And this was a multi-centric collaboration, and this whole concept of collecting samples, storage, transportation, all of it was taken care of to give the kind of results that was finally obtained. And to kind of come to the last couple of slides of the presentation, I think the present and the future is probably collaboration between the different biobanks. Now, there is a big, huge initiative in this condition, in this area, which is the Global Biobank Meta-Analysis Initiative. Now, they have this initiative has a collaborative network of more than 20 biobanks from 4 continents, and with more than 2.1 million consented individuals with genetic data linked to electronic health records. So if you see the map here, it covers almost all the continents and a number of thousands of patients recruited from different continents and different countries. So they are basically looking genome-wide association studies across the 6 ancestry groups. And the initial flagship project, they were looked at the most common as well as less prevalent diseases to see what are the genetic associations that can be studied. So this is probably the present as well as the future. And they have the future studies that have been, this was done about 2019, I guess, and now it is being continuing. And the group has been instrumental in identifying disease-associated genetic loci, gender-specific, ethnicity-specific, and they have shown that adding populations with diverse ancestries will improve the understanding of genetic variants, which impact disease as well as outcomes. So finally, to conclude, I think the biobanking has a great impact in terms of improving screening and diagnosis and treatment and clinical practice. It can provide better health and improve outcomes. It can provide new medicines and increase credibility and support for R&D. So it's a powerful resource for research and improving healthcare. However, the caveat is that it has to be accurately maintained in terms of sample integrity, ethical consideration, and data management. These are the keys for maintaining a proper and a well-managed biorepository. This is our team that has helped, and thank you so much for the opportunity. Thanks once again. If there are any questions, I can take it.
Yeah, thank you very much, Dr. Amritha, for a very, very illustrative and informative presentation. Just amazed, and I'm sure that you have threatened the audience who are just beginning to think about biobank with your the way the setup, the way you have collected the samples, the way you have done analysis, and of course you have followed strictly the guidelines at the same time. And every way, I think it's really a threatening message. But I'm sure that it's not a real threat as such. This is a volume of information and the volume of work you have, your team. It's not an individual, of course. It's a team work in place and the institutional support you get, and also the funding support through the research studies and from other sources. And I see the idea of bringing you as our first speaker, you have really delivered. You have really delivered, and I'm sure it will be very useful to everyone. And as from our principle of setting up biobank, and as every research institution, our dream is every research institution in this country should have a biobank. And yours is one of the model system where people should come forward to interact with you, and we will help everyone. whoever is interested to set up the biobank. And we have Dr. Abhishek Mohanty from SCG with us, and people like Dr. Abhishek Mohanty, Dr. Amritha, and we, of course, from Migene, we will help anyone who is interested to set up a biobank. We will support you in this process. And I would like to invite Dr. Rajinikanth Bangala, our founder of Migene, and a very amazing and interesting research person, and also equally an entrepreneur. I would like to invite Dr. Rajinikanth to say a few words, then we will take the questions from the audience. Over to Dr. Rajinikanth Bangala.
Thank you. Thank you very much, Dr. Elango. First of all, I would like to congratulate the speaker. It was Dr. Amritha. It's a great presentation. Fantastic work done by you and your team. Sincerely appreciate the amount of energy and the the kind of leader that you have become in making things happen there. Congratulations to you and to your team.
Thank you.
One of the main things that I have seen while I was the director of Thrombosis Research Institute in the same campus in Manzumdar Shaw Cancer Center. And at that time, Dr. Elango was also there in the main campus, and Dr. Jiny Nair was finishing her PhD almost 10 years back. Biggest challenge was the amount of electricity bill that was coming in storing these biospecimens. Either it is an electricity bill or it is the amount of liquid nitrogen needed. Lot of challenges, lot of manpower challenges, lot of financial challenges. And I give an example as to why this story of InstaPreserve started. In 2017 or so, the founder chairman of Thrombosis Research Institute passed away. And then there was no one to support the 15,000 patient biobank that we have established. And TRI had a blood samples, urine samples, tissue samples, name it, saliva samples, everything under one roof of 15,000 patients and a 10-year follow-up, a clinical follow-up. However, it could not be sustained because of lack of funding, lack of support in many ways, and eventually the biobank was completely removed. And that day was the biggest loss for a scientist like me and for any scientist. And that actually came up with a challenge that we need a simple, cost-effective, but better quality in biospecimen collection and storage. We really need to develop an Indian system, a system which is apt for any biospecimen collection and storage, and it should not involve costly -80 freezers or liquid nitrogen. And how do we come up with such a solution? That was the challenge, and this research of InstaPreserve started in late 2017. And from there on, quite a lot of research has gone in. We went on to come up with a solution called InstaPreserve, which right now is under evaluation with several of our partners, including HCG, Dr. Abhishek Mohanty, and he was kind enough to try our InstaPreserve. And we have established several collaborations now, both in three different aspects actually: human healthcare, plant, environmental, and animal. So this is the most unique thing, which is it cuts across the type of biosample. So InstaPreserve can be used in for storing human biospecimens. You can use in plant, animal, or environmental samples. We've even done soil microbiome. And in the video, if you have seen Dr. Vikram Diliyappa talking about human gut microbiome. So a kind of a platform which really addresses the needs of the scientists. You can store it at ambient temperature. You don't need a -80 freezer. You don't need a -20 or even 4 degrees. Such is the solution. Now, what we plan to do is to bring or build a society, a kind of a network, which would talk about what is the value of biobanking, as Dr. Amritha has spoken about, which is really very important, and how this value can be transcending across the scientific fields, especially in India and also globally. So I thank Dr. Amritha again, and Dr. Jiny, Dr. Elango, and also Ms. Sudeshna, who have taken a lot of pain in organizing this event. And last but not the least, I was just discussing with Dr. Abhishek Mohanty, and he has gladly accepted our invitation to be the next speaker in the next webinar, for which we will decide the date shortly in conjunction with him. Dr. Abhishek Mohanty is the program director and head of HCG Center for Biorepository and Biobanking. He is also the regional director of South India Biobank India Foundation. And we sincerely thank him for accepting our invitation. And I now ask the question and answers to be taken over. Thank you.
Thank you, Dr. Rajini. Here we have questions from the audience. The questions relevant to the biobank, I will pass it on to Dr. Amritha. And the questions pertinent to the InstaPreserve, I will take the questions and then I will answer it. And I think it should be fine. And the question from Raju Parak, it's the biobank governed by any regulations from CDSCO or DCGI? Dr. Amritha?
Sorry, Dr. Rengo, I just missed the question. Could you just repeat it?
Yeah, this is biobanking governed by any regulations from CDSCO or DCGI, Drug Controller General of India?
No, actually, as of now, I think the governing guidelines for the biobanking is from ICMR. So the ICMR, the 2017 guidelines that have been come out, they have included a section on the biobanking, and that is probably the governing guidelines.
Thank you very much. And there is a question from Kavita Sankar Narayanan. And the question is that would be that the product will be extrapolated to the current version to something like a solvent to dissolve nucleic acids, as well as could be stored at room temperature. If not yet done, please include this in our wish list. Thank you so much for providing additional thing. And as such, this product can be used for storing the nucleic acids. And if you contact us, we will provide how to use this for storing the nucleic acids. That isolated nucleic acids, I'm sure you have meant isolated nucleic acids can be used. This can be used for isolated Buffy coat as such, and then the plasma or the individual blood components can be stored. So if you contact us, whoever is interested in this product, we will provide a complete the profile of the different methods to store the samples, how the gel and the sample, there is a ratio where it has to be stored. We will provide a complete details to them. and also for the extraction. If you are storing the tissue, leaf, stem, soil, or even whole blood, if you are storing in the ISPA preserve, we will provide. The protocol is a little bit modification in the early stage. After that, it is the normal protocol you follow in your lab. So this as a very simple modification, which will help you to come out or customize it as per your need. So this product can be very much customized as per your need. They have someone as okay. Okay, Doctor. The message from Dr. Abhishek Mohanty is in head and neck cancer, it is a challenge to develop a stage-specific and subset-specific tumor bank, especially radiological staging and pathological staging needs to be documented. What's your comments about Dr. Amritha?
Yes, definitely. I think there is definitely a need for documenting radiological images, pathological images, and we are in the process of, we have initiated the program for doing that. And I mean, till now, I think we were focusing on the tissue samples and the biospecimen. But as I mentioned earlier, we are now looking into collecting all the necessary radiological images as well as histology, full slide images, so as to answer all the questions that is required. It definitely that is mandatory, I think now.
Thank you, Dr. Amritha. There's one more question from Raju Parekh is do biobanks need to be registered with regulatory bodies as ICMR, which I think you have answered it. If one more time, if you could insist, then that will be better.
Yeah, so there are certain guidelines that have been given in the national guidelines of that's released in 2017, I think, from the ICMR. We need to follow all of them. Basically, the requirement is that the ethics committee has to govern it in terms of collection, the patients who are being collected, the distribution of data, transportation of samples outside the institution. All of that has to be governed by the ethics committee as well as the technical committee that governs the repository. So this is what the ICMR guideline says.
That's good. And I have a question from my side. You have a biobank which is registered with the ICMR and you follow the guidelines. But if there is an international company or international your collaborative group, if they come and they for the research, even for kind of a pharmaco genomics, some information, if they come out and they try to use this biobank, and later they go for FDA approval, then how far this biobank is registered or recognized from a foreign regulatory authority like FDA?
So international collaborations, like for one of the studies that we mentioned, we have international collaborations, but that international collaboration was for sharing of image data, not for specimen. And that is governed by the HMSC, the Human Research Committee that is under the ICMR. So we have to get the HMSC clearance if you have to share any kind of data internationally. So it's only after that that the collaborative study itself can start. So we have an NIH grant wherein we are collaborating with the University of Arizona. So first they give us the proposal, the grant has been obtained. Then before we start the study in India, we have to apply for HMSC clearance in ICMR, which takes about 6 months or so. So before that, the ethics committee also does not give us our approval. You have to get that that HMSC clearance, and then only the study can be started. So after that is done, then the process gets streamlined. So even if it is goes for FDA approval, I think this should not matter.
Oh, that makes very clear. In that case, even if you establish a biobank, you go for a foreign collaboration, still you are under the governing body of ICMR, you get approval, then it is registered, recognized by the foreign authority also.
Yeah, so I'm not particularly sure about the biobanks, but for every project, any collaborative project for exchange of data, you need an HMSC clearance, which I think will be the same for biobanks also, with any kind of sample or data.
That's good. Okay. And yeah. Thank you, Dr. Amritha. There is one question from Ruby Gupta. Is related to in-stock preserve. How do you retrieve the samples like plasma or serum? Okay, good. See, when we mix the plasma serum with the gel, the insert preserve, it gets completely mixed and to the ratio. So when you take, you have to take, so suppose you have a plasma and the gel is 3 parts or 4 parts. So when you add the plasma, then 1 ml plasma and 3 ml gel makes 4 ml. So if you have to take 1 ml, you take the entire 4 ml and use it, which is representing your 1 ml plasma. In the first stage, only there is a difference where you have to use the reagents as per the 4 ml. Once the first stage, the gel is removed, then you can follow your entire protocol based the reagents for the 1 ml plasma. So which will be clearly given in the protocol when we provide to you. When you contact us, then we will contact you, will give you the complete picture about how to collect the samples and use the insert preserve, what is the ratio for every sample. We give a complete picture. Then for retrieval or the downstream process analysis, we give a complete protocol solutions to you. And any point of time, it's all can be customized. Okay, again, thanks for Raju Parekh asking more and more questions, keeping us alive. And I am part of a pathology lab. I get requests for the biospecimens. We don't indulge in these. Okay. What is this? The question is not yet clear. Okay, in the pathology lab, right at present, we have taken initiatives to work on how this insect preserve can be used for pathology specimens. Soon we will get back to you with details about, even we have got data from one of our collaborators that the insect preserve is very much suitable for keeping the samples and later taking it for histopathology analysis.
If I may actually, Mr. Raju Parekh is suggesting that maybe people do sample collection without ethics approval or something. So there are two types when it comes to pathology labs. You can use InstaPreserve as a medium to collect the sample and ship it to your lab. You can do the diagnostics and then you can, throw away the sample. If you don't have the ethics committee approval or ICMR regulatory follow-ups are not there, then you don't need to do biobanking. But the advantage of using InstaPreserve in sample collection and transport is that everything happens at room temperature. You don't have to process it, put it in cool packs or dry ice. Such a shipment problem won't arise when it comes to pathology lab chains, which can use our InstaPreserve for diagnostic purposes only. I hope I have understood. Yeah. So I hope you have understood, Mr. Raju. Yeah. Thank you.
So I really thank Dr. Amritha Suresh once again for being our esteemed speaker for this webinar, and it gave a very good beginning. And I thank the participants for sharing your views and asking the interesting questions. And I thank one and all, and I thank our team, Dr. Jiny, Sudeshna, for wonderful preparation and organizing this webinar. Very soon we will come back and meet you with our next interesting person, the interesting speaker, Dr. Abhishek Mohanty. Then followed by, we will have many personalities who are very much interested in research and who are active in biobanking will become our speakers. And I would request Dr. Amritha Suresh to come again with a case study in a biobank with a case study. So now you have given an overview, though it's an overview, but it was in detail. You have given about the biobank and the regulatories and the ways they have used. And if you could provide a case study how the biobank helped in the clinical research,
Indeed.
and that will be very useful. And we would like to have you again, Dr. Amritha. Soon we will come back and we will fix up a time with you. Thank you so much. And I thank one
Thank
and all
you.
for. Yeah, Dr. Amritha.
No, thank you, Dr. Lengo. It would be a pleasure to come back. But sure, let us know. Thank you once again for the opportunity.
Yeah, thank you so
Thank you.
much. We will see you in the
Dr.
next
Lengo, Dr.
webinar
Rajni,
soon.
meet.
Thank you.
Yeah, sure.